and welcome to this screencast where I'll be discussing the details and the mechanisms behind generating and maintaining the resting membrane potential in a neuron, as well as in, say, a muscle cell. But we're going to focus here on, again, the membrane potential. So by the end of this, you should be able to here describe what we mean by a membrane potential, and then explain how the membrane potential is established and then maintained in a neuron. So a neuron is a cell with a polarized membrane. So this term polarized, so if something is polarized, means that it is, or another way to think of it is that it is sided, that there are differences in that or of that, and that it is not uniform. These are other ways of thinking about how something is polarized. And so if the cell is polarized, and we said that the membrane is going to be the thing helping to keep something separated there. So the membrane is going to be involved with this. A membrane potential then is an imbalance. I'm reading this here. A membrane potential is an imbalance of charges across the membrane. Right? So a potential, you think of potential energy like water behind the dam. You have a difference in where the water is behind the dam, or on one side of the dam there's lots of water, on the other side of the dam there's very little bit of water. So this potential is an imbalance. And you can think of a potential as an imbalance, or that there's a separation of something. The resting membrane potential then exists in an undisturbed or unstimulated cell. So what we're saying is that the potential exists across the membrane. Right? So there is another way of describing this is that there is a difference across the membrane. And the difference in what? In the case of the neuron, it's a difference of charges. So the membrane potential is a difference in charges across the membrane. So if we have our neuron over here, we see our dendrites in our cell body, and then here's our big long axon and the axon terminal right here. And what we're doing is that with this device right here, this oscilloscope, we have these little electrodes. There's an electrode that's going to be inside the cell, and there's an electrode that's going to be outside the cell. So let's come down here. We'll zoom in on this. And so what we're saying is that we have this electrode right here, which is the measuring electrode. And this is detecting the charge inside the cell across the membrane. So this here is the membrane. And then this is the reference electrode outside the cell. Right? And so this difference in charges across the membrane is detected by this oscilloscope. So what we're saying is that the inside, this is measuring what the charge is inside the cell with respect to the outside of the cell over here. And so we're measuring that potential difference as a voltage. A voltage here is a separation of charges, or one way to think of it is the separation of charges. And so we have this millivolts here. Is very small levels of voltage, so thousandths of a volt here, millivolts. And what we're saying is that this oscilloscope is measuring that difference, and the inside of the cell is negative 70 millivolts with respect to the outside. So this is a comparison number. So the value here is how much charge is separated. What is the difference across the membrane? And that difference is going to be 70. And what we're saying is that since this electrode is inside the cell, then the difference here is negative. Because this is reading negative, we're saying that the inside is negative. Inside is negative with respect to the outside. Right? So the inside is negative with respect to the outside, or we're saying that the inside is negative compared with the outside with respect to, you could also say compared with. And this is a characteristic of not only neurons, but other cells as well. But specifically, we care about what's going on here in the neuron for this right now. So a characteristic of cells is that they are polarized, that there is a potential difference across the membrane. And it turns out that the difference is such that the inside of the cell is negative with respect to the outside, which is going to be positive. Another way to kind of think about this is that negative, or another way of describing negative, is that it is not positive, or that it is less positive. Right? Much in the same way that you have two sides of a coin, right? You have a coin is polarized, right? It has sidedness to it. You have heads and tails. Instead of saying tails, you could say heads and not heads, for example, right? And so all of these terms here sort of get used interchangeably when we're talking about this idea of a resting membrane potential or of an action potential, right? So this is the sort of the baseline setup for cells that are at rest. Right? A resting membrane potential is just this, that the inside of the cell is negatively charged with respect to the outside of the cell, and that difference is about negative 70 millivolts worth. Right? That's a lot of information. I think I'll stop here for this one, and you should review that and get your questions down, and then come back and watch the next screencast where I talk about how this is established. Why is the inside negative with respect to the outside? Okay?